Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing you not one, but two really awesome guests today involved in an area creating a better tomorrow for all of us. Today, we have the honor of being joined first by Marcie Finney, who is the Executive Director of the Cleveland Cord Blood Center, where her responsibilities include strategic planning, finance, administration, product development, and she has an extensive background in the area of cord blood. She spent over 10 years at Case Western Reserve University, participating in all aspects of cord blood research, and she's quite active in the cell therapy community, involved in sharing her knowledge, experiences with not just research, but also cell therapy and banking in the United States and globally. And the Cleveland Cord Blood Center is a nonprofit. It's an independent public cord bank serving as a leader in the collection, processing, storage, and distribution of these quality cord blood stem cell units for various aspects of transplantation in patients. Prior to this current role, Marcie served as an organizer of director of the hospital relations, where she headed education and training of collection coordinators and medical staff at hospital collection sites, has a master's of science in chemistry, did her MBA as well, Case Western Reserve University, and did an undergraduate in the chemistry as well as the Prague University. We also have the honor of being joined by Dr. Fran Verter, founder and director of the Parents Guide to Cord Blood, an organization that she started in the late 90s in the memory of her daughter Shai, who, when unexpectedly diagnosed with cancer, led her to really become a major patient advocate in this whole area. Dr. Verter started actually her career as a research scientist in the area of physics, got her undergraduate degree in Brooklyn College, did a PhD in astrophysics from Princeton University, and then ultimately found out that this really important training not only enabled her to understand what was going on throughout the cosmos, but really be able to understand and review medical research, to seek out therapy, to understand treatments that were in development. And we're really honored to have her. And she also launched as a subsidiary of that organization in 2017, celltrials.org, a really interesting system to help really collate and a really complete and accurate data set on all the clinical trials on advanced cell therapies that are going on around the world. Honored to have both of them with us. A lot of interesting themes to get into today. Marcie Finney, Dr. Fran Verter, thank you so much for both coming on the show.
Thanks for having us.
Thank you. I don't know if I can speak accurately as fast as you, but I am from Brooklyn, so I'll give it a shot.
Well, that's awesome. But yeah, let's start off with you, Fran, because obviously you have some very interesting background stories. Obviously not just with studying carbon 12 throughout the galaxies.
Wow,
but
you actually read my CV.
Yes. Yeah, I am extremely interested, not just in the life sciences, but in lots of other areas of science innovation. But yeah, Fran, take us into your story, because there's many aspects of it. Obviously, you're an astrophysicist, but you, as mentioned, you also are an advocate that made this area of cell therapy, of cord blood, a major second part of your life. And take us into your background story a bit, if you would.
Well, it just so happens that talking about myself is my least favorite topic, but everybody always wants to start with my story because my charity is founded in memory of my daughter. I was working as a research astronomer. I was employed at NASA. I was working on grant money, which is a kind of a freelance situation where you win a grant, you do the research, you publish the research, you get another grant. It's kind of a perpetual postdoc self-employment situation. And when my child was diagnosed with cancer and I started spending all of my time in hospitals, it disrupted that cycle and it ended up ruining my career. I mean, it really is true that if you do not publish, you will perish. And that's what happened to me in terms of getting more grant money. So I did everything I could to try to find the best protocol to treat my daughter. I learned how to read medical research articles. And I have to emphasize, nowadays people think that means using PubMed on your phone.
Mm-hmm. Right.
And in the 90s, when my first child had cancer, we did not have cell phones and we did not have PubMed. So when I was trying to find the best protocol for her, that meant going to the National Library of Medicine, which happens to be within driving distance of my home, and spending the entire day there. And you would submit a request for an article and you would wait for someone to bring it from the stacks. And then you would read it on paper and take notes. This is a very old school method of doing research. And I was copying down the names of the people who were primary authors on these papers, and I was writing to them, and some of them were, kind enough to answer me by email, getting more information. And this did, guide our treatment decisions. I was a very hardcore advocate for my kid, and I threw certain doctors off of her case and ended up taking her to a different hospital altogether. And she did live several years longer as a result of that, but I ended up losing my child and I lost my career as a researcher. So I came back to NASA in a job just as a computer programmer. Anybody who knows how to do physics also knows how to write code. So I came back, and when I was pregnant with my second child, I decided to bank the cord blood. And that ended up triggering my research into banks. And I discovered that the advertising didn't really tell you what they were doing. So I started phoning the labs, trying to find out what they were really doing, trying to get, somebody with a lab background to talk to me. And after I had compiled all that information, I decided, gee, maybe it would be helpful to other parents if I put this online. And that was how the Parents Guide started. It was really intended to be a hobby. And it gradually took over my life. I ended up switching careers and running a nonprofit foundation. Of course, along the way, I had to incorporate a nonprofit foundation, which is something else I had to learn how to do. I think a message I have for anybody whose career has been disrupted is that you can start over in midlife. And even if you completely switch, there are skills that you learned before that you can transport. And like in my case, the ability to read a scientific research article and interpret it is something I've been able to transport, even though I had to learn about stem cells and I had to learn all this terminology, but you can transport that skill. And if you're good at explaining science to the general public, then you can find work in a lot of different science disciplines. And so there were just a whole lot of core skills that I managed to transport. And, even to people who have no interest in cord blood, I want to have that message that just because, you're a single mother in your 30s struggling, to make ends meet, you'd be amazed what you can do with things you've done before, the skills that you have.
Mm-hmm. Mm-hmm. Yep, you look at a pharmacist that became a podcaster. So
there you go.
I can exactly. Here we go. Marcie, let's talk about you a bit as well, because you started off sort of studying chemistry, and again, I looked into your publication history here. You were not just working on umbilical cord blood, sort of in the early days, looking at things like neovascularization. You were studying ibuprofen and neutrophil migration, doing all sorts of interesting things with stem cells and sort of on the periphery of that as well. Tell us your background story, if you would.
Sure. So I went to Slippery Rock University, which is a couple hours from here. My dad had gone to undergrad college, but he did it after the military, so it was kind of secondary. So I was really the first one in my family to go directly to college. And Slippery Rock University is probably, it was a great place for me. And there were a lot of students there that were first generation college students. And I had a chemistry course. I thought I wanted to be a physical therapist, right? So I went there because of that. And I had a intro chemistry course, and I crushed it on my first exam. And my
Nice.
professor called me over and said, "How much did you study for this?" And I was like, "I don't know, like a little bit. It just makes sense to me, right?" So he said, like not everybody understands chemistry like that. He said, I'm normally everybody gets C's and then we work our way through. So he said, you really should think about this. And as I took more chemistry classes, I really, really liked it. So I ended up getting a chemistry degree, and I ended up working at Case Western Reserve as a research technician, where I did really analytical chemistry things. I worked on ibuprofen for cystic fibrosis, where we were looking at pharmacokinetics, because
Yep.
people with cystic fibrosis don't absorb like a normal person. So we wanted to make sure they got high enough levels. I was running GC mass specs and HPLCs. I also did some work with metabolic research, where we were also looking at analytes as for children that had metabolic disorders.
Yep.
And going to graduate school at the same time, right? My family didn't really understand how graduate school worked, so I couldn't afford to go to Case otherwise. So I worked there, took classes, was a bartender, and coached high school basketball at the same time. I don't know how I did all that, but um, I was working on that, and then it was complete serendipity that I got into cord blood. I had a girlfriend who was a drug rep. She was selling Neupogen to
Sure.
oncologists, and
Yep.
said, I met this woman who came to Cleveland from Duke. She's incredible. She's done all this cord blood transplant, and she's trying to start a program here at Case and is starting a lab. You should go talk to her. And I said, I can't. I'm getting married. I'm trying to get a PhD. I love my job. And she literally set up a meeting. I thought I was going to lunch with her, and I actually was going to an interview with Mary Laughlin, who is a pioneer of cord blood. She did the first transplant in an adult in the US when she was at Duke, and then started a program in Cleveland. And here I was going to an interview and didn't even know it, right? And I did decline the job, believe it or not, the first time. I said, I can't. I'm getting married. I'm taking my cumes. I'm trying to get done. So she said, I'll call you in a year. And I thought, yeah, right. This is never going to happen. But she did call me in a year. And I had some transformative things happen. I got married. I lost my stepfather unexpectedly to cancer quickly. He was only 52. And I thought, I think I want to try to do something with cancer. And Mary Laughlin called me again, and I left all of that analytical chemistry behind. And I said to her, I don't know anything about dealing with anything live. I'm an analytical chemist. And she said, well, you know how to run an experiment. You understand controls. You understand how to have a hypothesis and only change one thing, because you can't do that in an analytical experiment. She said, I think that we could take the biologists that know about cells, and you could teach them how to run a great experiment, and we could put it together. And I'll tell you, we really did. I had a mentor, Suzanne Ketteret, who was a cell biologist from Germany. We got a lot of stuff done. It was a great experience. And then here goes my life in cord blood. We then started recognize there was no cord blood bank in this geography. There was New
Yep.
York, St. Louis, Duke. So Mary Laughlin got funding from a private foundation to start the cord blood bank, and I went with her. And then I was part of all the recruitment of new donors and new hospitals. And then when our first executive director left, then I took over as executive director, I think 6 or 7 years ago. So I've had quite a journey. And when I got the job, I felt like I needed to understand business a little bit better. Just because we're a nonprofit, right? It's a tax
Sure.
status, not a business plan. So I went and participated in the executive MBA program at Weatherhead, where half the people were physicians, actually learning how to run organizations. So it's been an incredible journey. I'm really grateful.
Yeah, it's interesting. I'm here in downtown Center City, Philadelphia, and there's across the river a couple miles away is the Coriell Institute, which was one of the groups in this area that was kind of early on the scenes in terms of sort of cord blood blanking when it sort of first became a thing in this area. Love to have sort of and both of you, tag team on this one as you wish. Thinking back, sort of as I remember it, sort of the history of what we were explained, let's say, cord blood was all about was initially, the therapeutic potential of these stem cells for hematopoietic reconstitution, which then, as your research, both highlights, it was therapeutic potential of cells for more than hematopoietic reconstitution and various different indications. And then more recently, let's say, other bioproducts, because blood is not just cells, there's thousands of other factors that do things in there. Take some time, if you would, to sort of walk through the history of research in this domain and sort of where we've come from, say, the last 25 years or so, in understanding the potential of what this stuff, I'll just call it stuff, is in 2023.
Marcie, why don't you take that?
Sure. So I would say from my perspective, I think we spent probably that first 10 years trying to develop the techniques that we needed, right, to keep these cells happy, to isolate the correct cells, to make sure we're storing them the correct way. So I think that took some time, and it was a collaborative effort of a lot of people that actually talked to each other across the world, right, to figure out if, I think initially there was a lot of concern about the number of cells. Would there be enough cells in one cord blood to actually have therapeutic benefit? So I think that was probably the first step, right, to take that jump from children. Could we use them in adults? So I would say that was first, right, to really do a great job of honing that benefit for hematopoietic reconstitution. And then I think along the way, people started thinking, oh, aren't there other things, because they store so well, right? If you freeze cells the right way, my personal opinion is I think they're going to last a long, long time. I don't think that they're going to degrade too much over time. So I think people thought this is a great resource, right? There's no risk to the donor. It's cells that are completely thrown away. We've done a really good job of figuring out how to freeze them and thaw them so that they're viable, they work well. I think that first part was figuring all of that out. And then I think people started to think, what else can we possibly use these cells for, right? So I think thinking about cell therapy, and we did some work about vasculogenesis. Would they work for revascularization, places where you couldn't have an intervention? Spoiler alert, it did work pretty well, right? We didn't quite get all the way in my group, to cardiac uses, but they're out there now, right? And I think it hasn't quite, I still think that it hasn't quite, I think there's still unlocked potential in these cells that people haven't really thought about how best to use them. And I think as the cell therapy industry over here had been thinking about using autologous cells,
right,
I think now that some of that is going to cross over to, why couldn't we use these newborn cells? They have no exposure to any environmental issues, right? Maybe in the womb, but if I needed CAR T cell therapy, you'd use my cells. Lord knows what I was exposed to in my life, right?
Right.
So I think there's going to be some crossover. I think we still have a huge amount of untapped potential in cord blood units. There's 800,000 stored around the world, right? And right now they're really not, they're being underutilized is grossly right now. Mm-hmm. So I would say that is my take on it. And I think the cord blood industry now is trying to work with developers and think about new ways that we might use them, because logistically they're frozen, they're already qualified, they're already tested. We could send them out with 24-hour notice if we need to. So I think we need to really do innovate together to think about ways where these cells are going to be most beneficial going forward.
Mm-hmm. Mm-hmm.
This is Fran. Let me add a few numbers to a few of the things that Marcie touched on. There are labs that have been storing cord blood for decades, and it's been proven that cord blood cells are viable after 27, 29 years. So we're almost at the 3-decade proof mark. And that's just a laboratory test of viability. in terms of defrosting cells, thawing cells actually, and using them in humans, there's a public unit that was used for a transplant after 21 years. And there is a private unit that was given back to the donor after 19 years, both cases successful. So it's not just a case of, if you don't use it in a few years, you might as well throw it out. That is not true. They are potentially viable for many years. And the public banking system around the world actually has over 1 million cells in storage, and private banks have, in my estimate, maybe 9 million, 9.5, a lot. I mean, at least 9 million in storage in private banks around the world. So there are a lot of, people who could potentially use this if more applications were rolled out. There are certain segments of the medical community that are just really conservative about this, and I don't think that they are tracking the research, or else they'd be more excited.
Right. Fran, talk a little bit more about this, because you've been publishing extensively as well in this space, and some of your you did this review a couple of years ago on the first decade of advanced cell therapy clinical trials using perinatal cells. You also written papers about the probabilities that Israeli clinicians will use the cells. There was an interesting one here on stuff you're finding out abroad on sort of cell therapy regulations in China. Putting aside sort of the therapeutic, sort of the advanced therapeutic potential, we'll get more into that. But what else are you finding out as you're out there, as you're creating these databases of what's happening, not just for the clinical trials, but to educate, here's a patient in the United States, here's one in Brazil, here's one wherever. What else are you finding out that you can sort of give us the top line of some of this research?
Wow, that's like a pretty open-ended question. Well,
yeah, take your time.
So one of the things I've been presenting recently in talks, so my team did a summary of clinical trials using cell therapy for coronavirus, because we've been tracking all of the cell therapy trials worldwide for COVID. And we discovered that about 40% of these trials were using cells from perinatal origin, either cord blood, cord tissue, placenta, some combination of the above. But another thing that we noticed, which we think is important for a public health perspective, very often the cells came from really close to where the patients were treated. We're realizing it's very important for there to be local biobanks in countries around the world. COVID was actually kind of like a great equalizer, because a lot of countries that are not famous for cell therapy jumped in and tried to use cells to treat COVID. They just went to whatever, cord blood bank or any kind of institution they had nearby, and they tried things that, ordinarily people would have had a much longer pipeline before they let them run a trial. So we found that 30 countries ran clinical trials, and about 2/3 of these trials, no, more like 3/4 of the trials were using cells from donors. So this shows you how important it is to have biobanks everywhere, because we're bound to have another pandemic. It's something that people don't like to talk about, but we're living in a world of more vicious pathogens, more easily crossing borders. And in another pandemic, if borders are shut down and you want to do cell therapy, you need to have a biobank nearby. So one of the things we were seeing is that it's important to democratize that access, to have biobanks everywhere.
Absolutely. Marcie, talk a little bit about, obviously Cleveland Cord Blood Center, as mentioned in your bio, you do obviously the banking and the storage and all the other important components in terms of how we process and distribute and so forth. You're also quite active on collaborations, and I'd love to whatever you can make. I know there was an announcement last year on a company called Verter Therapeutics that you were working with. Again, thinking about sort of the multi-potential nature of cord blood and what's possible there. Talk a little bit about sort of what goes on a daily basis beyond the storage of these materials at Cleveland Cord Blood Center. A little bit about what you look for in collaborators, and then also, because just talk about how you're obviously there's a natural synergy between what you do and Fran's group. How do you both collaborate? If you can talk a little bit about you two working together. Obviously, it's quite synergistic. If you could both speak to that, that'd be cool as well.
Sure. I think probably the best way for me to kind of give you so when we were founded, we made a couple assumptions, right? We thought that our distributions would be based upon the size of our inventory. So we really worked to grow our inventory to 10,000. We thought that was a good manageable number for us. And we also wanted to make sure we had units with high cell numbers because that's desirable by transplant physicians. And then also I think about it on the other axis, we want to have one from a lot of different diversity, right? So our particular cord blood bank has collection sites in Cleveland, Atlanta, Georgia, and in the East Bay in California. So we have donors from across the country, much to Fran's, last point, so that we as a bank have donations from across the country. Spoiler alert, right? At the same time, the usage of cord blood was going down at the same time. So our distributions got to our projections 1 year, right? And then we recognized probably in 2018, we had this amazing inventory. We are we're 52% Caucasian and 48% other ethnicities. So we're really proud of that we have been, which is better than the actual population. So we've worked hard to have ethnically diverse units in our inventory. However, it didn't actually move the needle on distributions. So we recognized like in 2018 that we have this amazing inventory that was underutilized. And what were things that we as a public cord blood bank could do that would help to make that inventory more valuable and make it more better utilized? So we did a couple of things. One, we really worked on trying to utilize the units that come into our door every day, right? We were throwing units away, right? Because you want to bank only the highest ones, but what do we do with the rest of these? I believe we have 50 different material transfer agreements with different institutions. Nice. From academic researchers all the way to, clinical trials and distributing units to those that actually treat patients. And we've done work all along the way. So what this academic researcher needs in the beginning of their studies is way different than someone that is embarking on trying to get into a clinical trial. So they need a unit that looks and feels like what a clinical grade unit might be, but they can't afford to pay us for that for research uses. So we've tried to manage our inventory so that we are using every kind of unit we have and making them available to people for use. So I think that's one thing. The other thing we did was we established our own research and development laboratory, and where we are looking for things outside of hematopoietic reconstitution, right? We've investigated, can you use cord blood for Parkinson's disease? We are isolating iTreg cells from cord blood, and we think that that will be probably helpful for GVHD. We're looking at it for AMD, for macular degeneration, probably other types of autoimmune disorders. We've also looked at, can we isolate the monocytes from umbilical cord blood and use that in wound healing, right? We are seeing some indication that it is healing faster and a little bit of less infections with the mice that we use cord blood cells to try to heal their wounds. So that was another thing we did. And I think the third thing we did was also, we recognized that making that jump from research and development into clinical manufacturing is a really challenging jump, right? Researchers are used to making things on the kitchen table, right? So they are changing things every day. They're not worried about the external things, and they shouldn't be, right? But when you start to think about GMP and clinical manufacturing, really the environment is what matters, right? You have to have the appropriate HVAC. And we have an incredibly amazing and dedicated staff. When our bank got our BLA license, they're highly skilled, and there are only so many people actually in the world that know how to make an approved commercial product, and we have a whole staff of them. So we thought, how can we take that knowledge and parlay it into helping other people get in that pathway? So we built a separate lab across the hall, eerily similar to the one we have where we manufacture our clinical grade product. And we thought, let's help developers do that first step of manufacturing. So we have the environmental monitoring, we have validated all the freezers and refrigerators, we have all of the things around the product manufacturing that a developer would need instead of building their own facility. Much like you said with Devera, they have other places where they manufacture products, but they're starting a new line. So we can help them in that development phase where you're changing things. We have agility to do that. You can't go to a big CDMO on your first runs. Well, you can, or you'll be broke quickly, right? So we have the ability to be a little more flexible, and we have a staff that knows how to do all this. So those were the things that we thought, if we could help anybody get into that path, that clinical pathway that's using cord blood as starting material, it's just going to be a feedback loop to our inventory in the long run.
It's really interesting also hearing you as a chemist and thinking about CMC and traditional pharmaceutical development, talk about biologics and the complexity that goes into standardizing, as you were saying, all aspects of this development. Really, really interesting. Sorry, Fran, same question back to you. Tell us about how you collaborate, any interesting, whether it's Marcie or other interesting collaborative work that's going on.
I try to have my finger on the pulse of trends around the world, which involves a lot of professional gossiping, and then trying to depersonalize the information so I can disseminate it safely. But there are some interesting trends going on. Some of them are difficult for the industry to cope with. I mean, for example, Marcie alluded to this, the cord blood transplant rate has been falling in Western countries, the US and Europe. Hematologists prefer to do haploidentical bone marrow transplants. It's become very, very popular. And so the usage of cord blood in adults has really gone way down in the West, at the same time that it's booming like crazy in China. So when you look at the entire world, we've got different trends in different countries. But for a bank that's based in the West, and you have, donors that are likely to match people in the West, it's a challenge to find a use for your inventory. And that's what Marcie was talking about, developing new therapies, is how they're trying to meet their challenge. One of the international trends that I have come across is that there's a big need for more cord blood transplants to treat sickle cell and thalassemia. This summer, the Lancet, famous medical journal, put out an entire issue devoted to sickle cell and hematology. It was done by a commission that they had organized. And it turns out that the prevalence worldwide of carriers of these diseases is 7%. These things are not rare at all. I mean, in the United States, people think of them as rare, but that's just because this is a very white country. And that's changing. Demographics are changing. We are having more and more mixing of racial groups, which is creating more and more dissemination of diseases that tend to trend in certain ethnic populations. And a lot of people think, gene therapy is going to fix that. Gene therapy is, now approved for thalassemia. It'll be approved for sickle cell soon. I mean, it is true that gene therapy works, but it's incredibly expensive and it's incredibly complicated.
Right.
And it's much easier to treat these patients with a cord blood transplant. And the Lancet is recommending that there should be more transplant facilities in what they call low and middle income countries, LMIC. 75% of sickle cell patients are in Africa, and 95% of sickle cell patients are in low income countries. So there's a big need to develop more transplant programs. It's just a question of geographically they're needed in places where they're not right now. So there is potential for transplants to grow. It's just a supply chain problem of, matching the needs to where the expertise is.
Right. Yeah, and it's interesting you bring up that point about gene therapy and, of course, gene editing and these exorbitant price tags nowadays, and the fact that, yeah, we can't keep our eye off the lower hanging fruit that is less expensive and may work just because these things exist. They obviously they have their place, but they're not going to be for everyone in the short term, and so that's a really important thing you brought up there, friend. I appreciate that. What let's go just a bit into the future for fun, because we've talked about obviously what's going on, what's happened, what's going on now. Fran, I don't know if you saw the episode we did with Matt Roberts from the Center for Advancement of Science and Space at the International Space Station. They've been doing a lot of stem cell work up on the space station lately. What do you want to see in the crystal ball? If you had sort of unlimited finances to play with here, where do you want a lot of this to go in terms of whether it's therapeutic application or just other exploration of
if there were unlimited finances, I would like to see more clinical trials of cord blood for neurologic conditions like cerebral palsy and autism.
Mm-hmm.
So in the case of cerebral palsy, cord blood has definitely demonstrated efficacy. And what's needed to get it a market approval is a phase 3 trial, which is trying to get underway. Supposedly some people have agreed to fund it. I'm waiting to see it actually launch, but, supposedly it's going to happen. But, it hasn't quite got market approval, even though there's proof of efficacy that cord blood does help children who have cerebral palsy. Cerebral palsy is not something that parents often think about when they are pregnant, because among full-term babies, it's only 2 in 1,000. But among premature babies, it's 2 in 100 or 2%, which is not really that rare. And it's a significant developmental problem for preemies, and it's therefore very helpful if we could have market approval to use cord blood for that. And that really leads us to start talking about autism, another developmental issue where cord blood is in clinical trials. And it has only shown efficacy for children who are young and high functioning. So that specific group needs to go through more clinical trials. But at the same time that these trials have been kind of in limbo due to funding issues and controversy issues, political controversies actually, at the same time that the trials are proceeding very slowly, over the past decade, the number of 8-year-olds who have autism has doubled in the past decade. It's gone from 1 in 68 to 1 in 36. So anything that could help should be investigated.
Marcie, same question. I neglected to mention in your, because I was zipping through the bio, that you also are on the advisory board of this Gamida stem cell company in Israel, which is doing some really pioneering things in terms of getting these products and bioproducts to market. Big dream indications. I mean, you mentioned CAR-T before, and some of the things that are happening there. Where else would you like to see things happening as we look out into 2024?
Sure. I think I would actually still back up just a little bit, right? I still don't think that we are using cord blood enough in hematopoietic reconstitution, right? There's some staggering stats about the number of people diagnosed with leukemia and the number of people that actually get treatment with a stem cell transplant. And Fran touched on it a little bit. There is a lack of knowledge, I think, in the clinical community. I'm not a clinician. I don't want to tell people how to treat their patients, but I think there are some places where cord blood could be really valuable. I had a personal story, right, where I was introducing a documentary at a film festival that had a couple of donor story or recipient stories about people looking for a donor, right? And in the lobby was a family from India who was doing a donor drive for their father who had leukemia and couldn't find a match, couldn't find a donor in the registry.
Mm.
And I said to her, I'm not a clinician, but have you asked about cord blood? She said, oh no, they don't want to do a cord blood. And I said, okay, well, I I I felt compelled to say, you are not going to find a donor fast enough to actually help your father. But it is a wonderful thing that you're doing this for the people after him. But I feel like, and he ended up dying because he didn't get a treatment. I don't know whether cord blood would have helped him or not, but I'll tell you, if that was my father, I would have drug him all over to try to find someone to help him, right? So I
Yep.
think there's still some miscommunications about when or where to use cord blood. It's not clear for people about when to do that. So I think that there's no connected messaging about when to use cord blood for transplant.
Mhm.
And some of the things that we do well are we can ship them with short notice. So if somebody needs one right away, we can do that. There are so many people that do a search and then never end up actually getting a transplant. So I think there's some room in there to actually do better, at least in the United States. I think Fran alluded to the fact that in Asia, especially, it's a little bit better. So I first want to say I would like to make sure that we're actually using cord blood when we should, and that people aren't dying waiting for a match. There should be a cord blood for everyone. Maybe it's not the first line, but if you have nothing else, I would have to say that there's got to be some ways to use cord blood better in hematopoietic reconstitution. Secondly, I would say, there's so many different areas. Fran touched on a lot of the cerebral palsy, autism. There is also work with HIV. If
Yep.
a patient gets HIV and also has leukemia, you can find a specific cord blood with the CCR5 deletion that will actually cure both their leukemia and their HIV. We work with the
Yeah.
Fred Hutch on that. It works very well, right? So I wonder if there's ways that we can do better with that. I feel like there are also so many cell therapy indications that as they move from an autologous or donation from the patient themselves, that we can use our cord blood inventory. Right now we have a lot of collection sites. We don't store all of them. I would love to see a place where we have a better inventory turnover, right? Where we're shipping out more units so that we can put more back in our inventory. And I think the cord blood banks do a great job. The product is excellent, right? Most of the cord blood banks have a BLA license. So, those products are the top tier of things that we can use to treat patients. And I just would like to see them be utilized more.
Yeah, I didn't. I actually we profiled was patient number 4, I believe, with the CCR5 Delta mutation, was the City of Hope patient. I didn't even think of that, but thank you for bringing that up.
Yeah.
That's a really interesting another application that no, that's great.
And I think one thing about the cord blood banks, right? So they're not biased to
Right.
the donors quite as much as an adult registry, right? So
Exactly.
today, the units that come to our bank from across the country, we get shipments every day from all across the country. They are an immediate snapshot of what the population looks like today.
Yeah.
So there's no lag, there's no bias, right? We try to have collection sites across the spectrum so that we have a nice snapshot of that population. I don't know what that might look like, tomorrow. We might be able to find some other mutation like CCR5 that we just haven't even heard about yet. So I think there is also a breadth of genetic material there that we haven't even thought about what we might be able to do with. And most of the banks, we're really thoughtful about even how we store it, right? So Fran talked about they'll probably be able to be stored for, who knows, at least 30 years now. So we store them in a bag that's split 80-20. So you could take 20% off without thawing the rest. We also do segments. So there are 200 microliter segments on each bag that we could thaw one to do some genetic testing and see what, if we're looking for something in particular. So I think there's a lot of information there, and there is huge potential with cord blood that we've scratched the surface.
Yep. Marcie, Fran, while I have you, I just have to ask because there's been a kind of interesting trend going on in the longevity, anti-aging community lately based on something known as parabiosis, where, along the way, someone figured out that when you infuse younger blood into an older body, there may be some rejuvenation events. Have either of you and your organizations been approached by folks that are trying to take advantage of this interesting anti-aging trend?
Hi, this is Fran. I actually wrote an article on that for my October newsletter, and I originally thought that it was a scam that I needed to debunk, but then I looked into it and I discovered that the research is real, at the very least in mice, and they're starting to run clinical trials in humans. But when you give blood from a young mouse to an old mouse, it reverses a lot of markers of aging temporarily. And temporarily is the key word in that sentence. So based on that, they're starting to run clinical trials in humans. And this is an interesting little loophole in US FDA law. They are using plasma from young people, giving it to old people. The reason why it's a loophole is that it's legal to sell your plasma. You can't sell your organs, but you can sell your plasma. So now there are these clinics popping up all over. It's like the early days of the stem cell clinics. There are these anti-aging clinics popping up, which is why I thought it was a scam, because there was so much commercialization. I distrusted it. But these clinics are giving young blood to older people. And, hey, who is the youngest blood donor? A cord blood unit. So there is legit research behind it. I think that in the short term, cord blood banks could possibly make some extra income by selling cord blood plasma, because normally they throw that out. In the process of spinning down the unit to get the stem cells and other cells that they're going to freeze, they actually spin off the plasma. So if you have proper consents from your donors, you could actually sell that, increase your profit margin. However, in the long term, I think that what will eventually happen is that more serious research will identify specific factors in blood, and perhaps cord blood could be used to manufacture those factors. And I have to emphasize that all of this is temporary. So personally, I have no intention of running out to get an infusion, because it reminds me of getting Botox and having your face frozen for a while and then it wears off. I mean, I don't see the point in chasing the temporary elixir of youth. That's my take.
I think it's a really interesting conversation, right? So as a bank, we have a responsibility to those donors, right, about what ends up happening to their donation. And we purposefully, our mission and vision and values of our organization were very much specific to transplant, right? To, reconstitution, the way all of our things were worded, right? And we purposefully redid our mission statement to include, it was all about saving lives, which is what you think about with transplant, right? Sure. But we recognized someone approached us about using cord blood for macular degeneration, right? So that's not really saving someone's life, but it would certainly enhance their quality of life, right? If it would stop them. And it was an interesting question that we ended up actually revamping our mission statement to include saving lives, enhancing health, and expanding knowledge, right? So that those are the things that we think about when we're trying to decide about a partner to work with. And as Fran said, there are some bad actors. It's our responsibility to make sure the people we're working with, and we have a big process that we go through legally and philosophically and regulatory space, right, to figure out whether they're people we want to work with in the long run. But we also think about, no one donates their cord blood to us for us to throw it in the garbage too. Right. So we have added a very extensive discussion in our collection consent form, where we actually talk to the donors about what are their wishes, right? So they can actually say, I want it to be used for transplant only, and if it's not that, please discard it. Or they could say, check another box that says you can use it for transplant, or you can use it for any other thing that you deem valuable. So we have that kind of an opt-in, and we have the discussion about commercialization, that we may work with a commercial partner. You mentioned Gamida before, right? So Gamida's commercial, but that means it's actually being a patient is being treated. They have gone
Right.
all the way through and got their BLA. So I think there's also some miscommunication about the language, right? Commercial is not necessarily a bad word. The partner might be, but if it's a commercial product, that means it's been all the way through the approval product and it's approved by the FDA. So we are continuing to investigate. We throw out plasma every day, as Fran said. So we're working in our lab to see if there are ways that maybe even the small units that don't meet the criteria to be frozen, could we isolate the plasma from that and save it? And we've actually had people also contact us about using it for serum-free media, right? So that it's not.
Right.
So I think there are many, many components of cord blood that we also have not really, we haven't utilized well. So we're investigating, and we'll talk to you next year about it.
Awesome. Very exciting time for innovation. Fran, Marcie, what is coming up for both of you that we should know about in terms of we're almost to 2024. Are there conferences that you're going to be presenting at, talks you're going to be giving, places that we can run into you, meet you as you both continue to create the awareness, the advocacy in this really untapped area? Please, both of you take the floor on this one.
Well, I'd like to return to the concept of myths and misconceptions that are persisting around cord blood. And we've talked a bit about the growth of the industry, and we talked a bit about whether or not hematologists are utilizing cord blood where they should. There's also the public aspect, because I'm running the Parents' Guide to Cord Blood, and I spend a lot of time trying to educate parents. And what is a big problem right now, it's both a plus and a minus, is the practice of delayed cord clamping, which has become very popular with parents and midwives to the point that they have a kind of a mysticism about it, that if you don't delay clamping and try to push all that cord blood into your baby, that somehow, your baby is not going to be healthy. And it's wonderful that all these people have decided that cord blood is important, but they have gone overboard with it, because delayed cord clamping is a situation where more is not better. A little bit of delayed clamping at the time of birth to allow some of the cord blood to go into the baby, a little bit is fine. But more than that, you're not going to get more cord blood into the baby if you wait a long time. The only thing that's going to happen if you wait a long time is that you won't be able to collect the cord blood. So we're having a problem with midwives in particular who want to wait, 2 minutes, a really long time. And this is important for both the public and the private banks. When a baby is born, you need to clamp the cord pretty promptly if you want to have a cord blood collection. If you want to have the best of both worlds, that you have both let some of the cord blood with that rich stem cell content go into the baby, but you also want to collect some of those stem cells for later use. And you can only do both if you keep the delay to about 30 seconds. And that is something that I spend a lot of time educating about, and I talk to nursing groups, to cord blood educators. It's wonderful that parents, now believe that cord blood is, the elixir of life, but I wish they would have a more balanced perspective on it.
Got it. Marcie?
Sure. So we also have a lot of conversations with potential donors about this, right? And we talk to them about the fact that ACOG recommends, the American College of Obstetrics and Gynecology recommends 30 to 60 seconds, right, for delayed clamping. And if you think about what's actually happening, right, you have the placenta and you have the baby, right? There is always an excess of blood, right, flowing. You couldn't have all of that into the baby. It's not built to do that, right, just from a physiological standpoint. So ACOG recommends 30 to 60 seconds for full-term babies. And we talk to families about the fact that if you do that, you have about the same odds as you would to have a clinical grade unit, a unit that's big enough to actually treat a human, right? And we talk to them, it's kind of a continuum. The longer you wait to delay clamping, the less likely it is that it'll be a unit that'll be stored and be big enough to be stored in our inventory. We could still use them for research, but it is a long conversation that we have with a lot of donors. What I would love to see is a pediatrician weigh in on the subject about, sometimes I've seen cases where the baby ends up jaundiced, right? Because they have too much blood in there. So I think we really need some clear guidance from pediatricians about what to do with delayed clamping also, and the longer term effects of that for the baby afterwards, right? We don't ever want to interfere with people's birth plans or with, what the recommendations are, but I do agree that this might be a case where more is not better for the baby even, right? We don't know yet.
It's a really interesting area, I gotta say, and it's obviously with a lot of the areas of biomedical innovation we touch on the show, we have still a lot to learn, and it seems that you are both you've both focused on learning, but obviously at the same time in teaching everybody else along the way. So I take my hat off to both of you. Really amazing work. It's going to be interesting to continue to see what happens in the space, to follow both of your respective programs, but really just great work, and big fan, applauding both of you. For
I would
everybody,
encourage everyone to participate in World Cord Blood Day, which is on November 15th. I would also encourage everyone to take a look at, I'm on the executive committee of the Cord Blood Association, which does a lot of work with both public and private banks. And I think that their website is actually really helpful to people that need some information. Their big meeting is the Cord Blood Connect meeting, which is held every September. So I would encourage people that want to know more about cord blood. That's the place to probably get the most directed information. I also participate in a lot of conferences that work with developers. There's a new Donor Selection Summit conference that'll be in the end of November that I'll be on a panel there about using cord blood as starting material. And also I will go to Advanced Therapies Week in London in March to also talk about using cord blood as starting materials with developers in Europe. So there are a lot of opportunities to get information about cord blood, and we hope to see a lot of people participate in those things.
Excellent. And we will put those links. And I neglected just at the time, but to mention that you are on the board of directors of the Goodwill Association. But we will put those links in the bio as well when we publish the show. Again, for everybody that is going to be listening to this particular episode of our show across the podcast networks, or watching on our YouTube channel, you've been spending time with Marcie Finney, Executive Director, Cleveland Cord Blood Center, and Dr. Frances Verter, Founder and Director of the Parent's Guide to Cord Blood.
And let me throw in a plug. My website is parentsguidecordblood.org. Okay, got that obligatory plug in.
And celltrials.org.
Okay.
But Marcie, Fran, I again, I want to thank you both for taking the time out of the schedule to come talk about these topics with us today. Obviously, thank you for both of what you do, and as we like to say on our show, thanks for helping to create a better tomorrow for everybody via the work you do. Really great having both of you.
Thank you, Aura.